components like lymphocytes and cells were discussed last time. Today we'll see how they create an immune response. So what are the steps that those components, cellular components, they create an immune response. The learning objectives for this session include: describe the generation of effector and memory T cells and its clinical significance, explain how helper, cytotoxic and regulatory T cells contribute to cell-mediated immunity, explain how CD8 positive and natural killer cell mediate cytotoxicity. Okay, here is if an antigen or an allergen enters our body, what's going to happen? An immune response will be generated as long as this antigen is from a pathogen, whether a bacteria, virus or a parasite. Do you think this word antigen is correct here?
No.
So one of your friends say no. What creates an immune response? Excellent, pathogen. But what is the technical term for pathogens? Yes.
Complex protein.
Do you all recall immunogen? Immunogen is an antigen that can generate an immune response. So we put here antigen to trigger that. Antigens can bind to the receptors of the adaptive immune system, to antibodies and to T cell receptors and B cell receptors. But immunogen for sure causes an immune response. Because we are discussing that how an immune response is generated, so best was if we had put it here immunogen or allergen. Now immune response can be from the innate immunity and from the adaptive immunity. Innate immunity we have already discussed. The adaptive immune system, if the T cells or the create the immune response, that will be termed as cell-mediated immunity. But if the B cells through antibodies create an immune response, that will be termed as humoral immunity. What does the word humoral means? Sorry? Fluid or soluble. In the fluid, something which is soluble. So antibodies are soluble, that's why it is called as humoral immunity. Do you think humoral immunity alone is enough to eliminate the infection?
No.
Sure, it doesn't work alone, it has to work with the other component. So now we'll just briefly compare the humoral versus cell-mediated immunity. One of the major differences is humoral immunity, it is against the extracellular pathogens. Now that extracellular pathogen can be bacteria and can be virus, while cell-mediated immunity primarily works against the intracellular pathogens. That can be intracellular virus, bacteria and also tumor antigens or tumors. The humoral immunity, it is generated or produced by the antibodies, while cell-mediated immunity is through T lymphocytes and it is through direct contact. We'll see all these things one by one. So this is the focus of our lecture today, cell-mediated immune response. Now what components are involved in cell-mediated immune response? T cell. Which other cell you expect? Dendritic cell. Natural killer, very good. Macrophages. So macrophages, natural killer cell, dendritic cell and T cell of course. And what components of the T cells? CD8, which is against the viruses, cytotoxic T lymphocytes, natural killer cells, then the helper T cells, regulatory T cells, they all will work. We'll see today all of them one by one how they are going to perform the function. But first thing is how the antigen will be recognized. So the bacteria enters, let's say, we'll keep the example of the last lecture, that the boy who was walking and he had a nail prick, and let's assume that a bacteria and a virus, or let's say an intracellular bacteria and an extracellular bacteria both entered the body. So which cells will first encounter or recognize the antigen? Dendritic cells and macrophages. Through which receptors? PRRs, very good. They will recognize the antigen first. Then they should take it where? Lymph nodes. Macrophages will take it to the lymph nodes as well? Because macrophages do not circulate. They are just in the tissues. But which component of the macrophage can take the antigen? Monocytes. So monocytes later transform in, so monocytes are circulating in the blood. When they reach the tissue, they convert into macrophages. But macrophages, they do not circulate. So if it was in the foot, the tissue of the foot, it will just capture the antigen and stay there. While the dendritic cell take the antigen and move to the lymph nodes. Now, since we are talking about two assumptions, that possibly it's an extracellular pathogen and an intracellular pathogen, so that's why we have here put both the possibilities. The dendritic cell is going to present the antigen to CD4 cell for extra or intra? Extracellular pathogen. And dendritic cell will also present the antigen to CD8 cell for intracellular pathogen. Now, for the activation, what three steps are required? Number one is MHC class 1 to CD8 and MHC class 2 to CD4. That's number one. Number two was co-stimulatory signals, B7, CD8 T-86 or CD28, and then third step was cytokine production. And as a result, what will happen? Differentiation and proliferation. So in the second phase, we have got now effector T-cell against the extracellular pathogens and effector T-cells against the intracellular pathogen. Now this has been produced in the lymph node, or let's say, because we are concentrating on the previous example, so maybe in the lymph nodes of the leg. So they have to now, the activated or effector T cells and effector B cells, they have to go back to the site of infection where the nail entered the body. So this needs migration of these cells, effector cells. So this, you have the figure here, these cells have to go back to the tissue where macrophages have engulfed the pathogen and they are waiting there. And now the effector function will occur. So the cells have to be killed or the pathogen have to be killed. Which will kill? So the intracellular pathogen, whether it is a virus or a bacteria, that needs to be killed because the virus is inside the cell. If you want to kill the pathogen, you have to kill that cell. Okay, so this job will be done by which cell? No. So we have now two examples going on. On one side we have got intracellular pathogen, which is dealt by MHC class 1 and CD8. Is it clear? So for example, oops, sorry, let me. So this one is the extracellular pathogen. Okay, the extracellular pathogen is presented here and it is dealt by CD4 cell. The intracellular pathogen is here, it is presented to CD8 cell. Now on this side we are thinking about the extracellular pathogen, on this side we are thinking about the intracellular pathogen. Extracellular pathogen, CD4 is first activated by 3 steps, differentiated. That full group, they have to migrate to the tissue. So migration is very important, and then they reach the place, the tissue, where they have to perform the function. The other side, CD8 cells, they are activated, effector T cells have to migrate. Now they both come here in the tissue. So there is cell, let's assume that this cell has got virus or this intracellular bacteria inside. So here the macrophage has engulfed the extracellular bacteria. Here the intracellular bacteria is inside the cell. Macrophage stay in the tissue, the effector CD4 cells come here and CD8 cells come here. So now what we will see, the effector cells migrate to this part. Here comes CD8 cell, and which cell should come here? CD4. Now they will perform their effector function. What is going to happen? They will perform the killing mechanism. We'll see about that, but before that, another thing you have to remember, I told you that the B cells, they also are the antigen presenting cell. They can also recognize the pathogen. So let's assume that the extracellular pathogen was recognized by B cell as well, and B cell is here in the tissue as well. Actually, there's no duster. So we'll see three different components here. One is B cells, one is CD8 cells, CD4 cells. This is extracellular bacteria, and he is dealing with a cell which has got a bacteria inside it. So now they are all effector cells. They have all been activated in the lymph node. All are here in the tissue. This B cell will perform activity against the extracellular bacteria by producing antibodies. This CD4 cell is going to kill this with the help of the macrophage. Sorry. So this CD4 is going to activate the macrophage. This CD4 is also going to activate the B cells. And this CD4 is also going to activate CD8. That's why it is called as helper T cell, because it's helping everyone. It's helping B cells. What is the purpose of this help? Help the B cell to start class switching. Instead of IgM, they will start producing IgG, IgE, IgA, depending on what is needed. Then CD4 will tell the CD8 cell, kill efficiently. We'll see what are those steps. Then CD4 will also tell macrophage to kill efficiently. All these effector killing mechanisms we'll see one by one. But here what you see in this figure is that B lymphocyte recognize the antigen, proliferate, differentiate into what? Antibody producing cells. Then the helper T cells, they recognize the antigen, proliferate and differentiate into CD4 cells or helper T cell, TH1, TH2. All this is going to happen one by one. Now, if we compare three different types of cells, naive cells, effector cells and memory cells, because these are the stages. You saw the first cell that came out of the thymus was naive mature T lymphocyte or naive mature B lymphocyte. These cells then become activated in the lymph node, and then finally effector cells, and in the end when the job is done they become memory cells. So what are those basic differences between these three, whether it is a B cell or a T cell? First thing is, if you look at the lifespan, naive cell has got weeks or months. That's the lifespan of a naive cell. Then the effector cell, it is for just few days. Why? Because its job is just to kill and that's all. Then it has to just vanish. And finally the memory cells, that has to stay longer in the body. So that's why its lifespan is for years. Effector functions. Naive cell, do you think it has any effector function?
No.
Because it is just waiting for the antigen to come. Effector cell, yes, it has got lot of functions. Helper CD4 has its own function. B cells have to produce antibodies. Memory cells, what is its effector function? It will again become activated when the antigen enters the body, so no effector function. Again, naive is waiting stage, memory is waiting stage, and effector is the main functional stage. Then what kind of antigen they recognize? So the effector cells and the memory cells, especially for the B lymphocyte, they have to become very mature. There is a special concept of hypermaturation, somatic hypermaturation, that will be discussed tomorrow. So just at the moment, they recognize in the beginning, what I will tell you, the one-liner is that in the beginning they recognize it very weakly, but by the end of somatic hypermaturation, the binding affinity is very strong. They recognize the antigen very well. How it take place, that in tomorrow lecture. Finally, the migration. Naive T lymphocytes have to go to the lymph nodes and come back in the circulation. They will keep on doing that. They will keep on circulating like this until they find their antigens. While the effector T cell, it has to go to the tissues. So once it is activated in the lymph node, it has to immediately go to the tissues to perform the function. While the memory cells, they can stay in the lymph nodes and also in the bloodstream. Now we'll focus on this part, that CD8 cell has to kill the cell in which there is a bacteria, intracellular bacteria, a virus, or any stress situation. It kills by 3 mechanisms. Okay, so this killing of the cell which has got the bacteria or the virus is by 3 mechanisms. Number 1, they come close to each other, and this CD8 cell is going to release certain special enzymes that are called as perforins. And the word perforin means perforate or make holes. Okay, so their job is to act on the membrane of the cell in which there is bacteria and make holes. And what will happen, of course, the fluid will go inside, the electrolytes come out, so electrolytes and fluid imbalance as the cell rupture or lies. So that's the first step, that's the first mechanism. But before that, of course, CD8 is going to respond to MHC class 1. So all nucleated cells, they have got MHC class 1. Whenever the virus comes in there, they can take that virus, present it with the help of MHC1 to activated CD8 cells that have come from the lymph node. Now they recognize each other. Co-stimulatory molecule is the second, and the third step is the cytokine. Here in this case, they produce perforins. Okay, those perforins will make holes in the target cell. That's number 1. Number 2 mechanism is they can also produce granzymes. So the cytotoxic T lymphocytes, they have got perforins and they have got granzymes. They are the enzyme that will trigger apoptosis. You all are familiar with apoptosis? So programmed cell death. They will cause kind of suicide of these cells because they have got virus or they have got intracellular bacteria in them. And number 3 is a special, FAS ligand, FAS ligand. We call it death receptor. Okay, so death receptor will bind to the FAS receptor on normal cells, and this expressed because of the intracellular pathogen, the bacteria or the virus. So this again will trigger apoptosis. So now just very simply, number 1, you make holes in the cell. Number 2, you trigger apoptosis either by the help of the granzyme or by the help of the FAS ligand receptor interaction. Okay, or the death receptor interaction. So ultimately what you want is death of the cell in which there is a virus or there is a bacteria. So far all of us are okay? Okay. Yeah.
What does the granzymes exactly mean?
Granzymes, they go inside, they are going to trigger, so you know, whenever anything binds to a receptor, there is second messenger system, they will go to the genes and trigger, you know, programmed cell death. So they play with the transcription factors inside. So they go to the nucleus and tell that there is no need of you, you need to destroy yourself. Trigger suicide inside those cells. Death receptor, Fas ligand. You just remember Fas, L is for ligand, Fas. So on one side will be ligand, on other side will be receptor. Just remember it is a death receptor and end result is apoptosis. So two mechanisms for apoptosis, one is for making holes. Now, can you name the non-specific cytotoxic T lymphocyte, a cytotoxic lymphocyte? Non-specific. Natural killer cell. Very good. It is a non-specific because it basically belongs to the innate immune response. Now, there is a very good complementary function between natural killer cell and cytotoxic T lymphocytes. So here you can see that the cell, it is then the natural killer cell comes and recognizes it and then it kills it. So again it is also initiating apoptosis in the virally infected cell, but the mechanism is slightly, the initiation or the method they follow is slightly different. What is that? So before I explain this, I'll tell you one thing, that bacteria and viruses, when they go in our body, they are not just sitting like that. So if our immune system is trying to tackle with them, they also have some tricks. So there are jumping genes, they can mutate, and that's why, and also, you know, this antibiotic resistance. The reason for this is because they are also playing with our immune system somehow to hide, somehow to protect themselves. One of the mechanisms that they have learned is that they know that when the intracellular bacteria go in the cell, MHC class 1 will present the antigen. So the viruses, what they do, they go in the nucleus and switch off the gene for MHC class 1. So MHC class 1 cannot express. So what cytotoxic T lymphocytes now cannot recognize it, but the nature has an alternate plan B for them. And what is the plan B? Natural killer cells. So whenever there is a MHC class 1, whenever natural killer cells see MHC class 1, he is quiet, he will do nothing. But whenever MHC class 1 is off, you know, the cell is not normal, first thing is that it has to have either virus, bacteria or tumor. So the natural killer cell knows that there is something wrong, but MHC class 1 is not expressed. So when there is no expression of MHC class 1, natural killer cell become activated. So the triggering factor is no MHC, so it's a balance. Cytotoxic T lymphocyte recognize MHC class 1, virus switch off that MHC class 1, natural killer cell recognizes it. So they balance each other. So in this figure you can see that there is a missing self-recognition. So MHC class 1 is lost because of the viral trigger, or that is one method of recognizing. The second method natural killer cell recognizes the cell is because of the stress. So when there is a tumor in the cell, the cells will express strange kind of receptors. We call them stress receptors. Those stress receptors will also activate the natural killer cells. So what if the MHC class 1 is not expressed? Natural killer cell will take over. So if, yes. Sorry? Stress receptor will be activated whenever virus sits inside and create, you know, production of viral proteins, or the cell has got now tumor inside. Again, in both the conditions there are a lot of proteins produced by the normal cells. Normal healthy cells, they have a check and balance how much protein to produce, but when virus comes inside, they create lot of chaos, viral proteins are produced, so stress is produced. Tumors, again, lot of unnecessary proteins are produced, stress is created. So in both these situations there will be expression of stress receptors. Again, there will be a tumor immunology lecture, so we'll discuss that part later. So now we'll see the recognition is slightly different. So you remember now, what were the three mechanisms of cytotoxic T lymphocyte killing? Granzyme induce apoptosis, perforins make holes, death receptor fast ligand is also going to initiate apoptosis. Cytotoxic T lymphocytes, natural killer cells, they have slightly different method. They have got receptors for immunoglobulin G1, IG1. Okay, do you all recall the parts of the antibody? This was called the variable portion and this was called the constant, and that's why this part was called as FC and this was called as FAB, antigen binding fragment. So FC portion, and if you remember, I told you this part binds to the antigen and this part performs the function. Yes, so how it will perform the function? So let's say this is the natural killer cell. This is the natural killer cell and it has got receptor for, and this is immunoglobulin G. So the receptor, this receptor is called as FC, FC from this part, FC, gamma, gamma is from this G chain, and R is for receptor. So it is the receptor for FC portion of the immunoglobulin. Does it make sense? That this is immunoglobulin G and this portion is FC portion. So naturally the receptor that will recognize it is called as FC gamma R. It can be 1, 2, 3, there are different types. So FC receptors are present on natural killer cell. So when the the bacteria comes in the body, intracellular bacteria, the antibodies, they will bind to the bacteria. So you can see this bacteria, it is recognized by these immunoglobulins, which are IgG. Okay, once these immunoglobulin recognize it, its FC portion is now ready to bind to the natural killer cell. And it has got FC gamma R1 receptor for it. Once it recognizes it, so this is also called as opsonization. You remember I told you complement will bind around the bacteria or antibodies will bind around the bacteria. It will be phagocytosed by the macrophages and the neutrophils. That is opsonization or phagocytosis. But here they tag the bacteria for natural killer cell to recognize. What will happen next? The natural killer cell will release toxic enzymes into the bacteria or the cell that has got the bacteria, and as a result that cell will be destroyed. So this is also doing cytotoxicity but with the help of the antibodies. That's why it is called as antibody dependent cell mediated cytotoxicity. So if you look at this word in detail, antibody dependent, it means if there is no antibody, natural killer cell cannot recognize it. Cell mediated, so the natural killer cell will be activated to do what? To kill the bacteria or to kill the cell which has got inside bacteria. Okay, so ADCC is the short form for this.
Same enzymes.
Yeah, perforins and granzymes, they can release, but its method of activation is different. So I'll just repeat again. Cytotoxic T lymphocyte, they will produce the apoptosis with the help of perforins, granzymes and fast ligand, death receptor. But this natural killer cell, it will recognize the intracellular pathogen, one, by the loss of MHC class 1, or number 2, with the help of the antibodies. That's why it is called as antibody dependent cell mediated cytotoxicity. Now it's a good time to explain one more thing, that this extracellular bacteria, or intracellular bacteria, it has got IgG antibody against it. And I told you that the first antibody that is produced is IgM. Okay, it start producing IgG against this bacteria. How? Because this CD4 cell helped the B cell to class switch. So they all work